I'm glad you could join me today. Today I will be going over variation and the genetic and environmental factors. So this is OCR Biology Spec 6.1.2a. Alright then, so here we go. Variation can come in two different variations. Discontinuous variation. A characteristic which can only appear in specific values. The individual either has the characteristic or they don't. It is controlled by a single gene, so it's monogenic, or a small number of genes which in turn have a large effect on the phenotype and interact in an epistatic way. So this type of variation the variations seen in this include dominance, codominance and recessive alleles. The environment will have little effect on these genes. Continuous. A characteristic which can have a wide range of values. For example, height. This is the combined effect of many genes, so it's polygenic. And polygenes are found on different chromosomes. Now, continuous variation is affected by both genetic and environmental factors. The causes of genetic variation. The obvious one of mutagens. You can have chromosome mutations, so deletion, part of a chromosome is lost. Duplication. Part of a chromosome is duplicated, which then could lead to overexpression. If overexpression occurs, too many proteins would be made, and that would cause our metabolism to be disrupted. Inversion. A section of the chromosome breaks off, turns 180 degrees, and joins again. So this means that some of the bases now on this chromosome are now going to be too far away from their regulatory nucleotides. You can also have translocation, where a piece of the chromosome breaks off and attaches to a separate chromosome. The final one is non-disjunction. Non-disjunction is when one pair of homologous chromosomes, or chromatids, fail to separate during anaphase, leaving a gamete with an extra chromosome. This can lead to things such as Down syndrome. Polyploids are organisms that have more than two sets of chromosomes in their zygote, and these are normally sterile. So, for example, if I had three, it'd be a triploid. The source of genetic variation. Crossing over in prophase 1, independent assortment of homologous chromosomes during metaphase 1, independent assortment of sister chromatids during metaphase 3, and a random mutation. Variation due to environmental factors. You've got variation caused solely by the environment, which could be phenotypic variation which is not passed on through genes, so speaking with a regional dialect, or losing a limb or having a scar from an injury or operation. Finally, variation caused by the environment interacting with genes. You don't need to know these examples I'm about to give, but it would be good for you to be aware of them. Chlorosis in plants. Leaves look pale yellow due to cells not producing the normal amount of chlorophyll. They could be down to lack of light, mineral deficiencies, or a virus infection. Finally, you got etidation, where plants grow in partial or complete absence of light. And finally, animal body mass. And that's everything you should know about variation.